Submolecular dysregulation due to deuterium accumulation is the cause of mitochondrial dysfunction. I quote, "Every chronic disease is linked to mitochondria now. The chronic fatigue, the cancers, you name it," stated Dietrich Klinghardt, MD, PhD, on the recent Human Longevity Project, a 9-part documentary about health. 80 to 95% of all chronic disease is the result of mitochondrial dysfunction. In this presentation, I will cover various aspects and functions of mitochondria, how metabolomics identified the mechanisms that detail submolecular dysregulation, how to reverse the dysfunction, and some future directions for research and understanding. Please know that this is for informational purposes only and does not constitute professional advice. Mitochondria are organelles that have evolved via symbiosis from bacteria and are found in all cells in the body except red blood cells. On average, each cell contains 1,000 mitochondria with higher amounts in some muscles and brown fat. Mitochondria can take up as much as 25 to 40% of the cytoplasm. They are highly dynamic and have metabolic and regulatory functions. They have an outer and inner membrane, and the infoldings of the inner membrane are called cristae, which house the OX-phos complexes. The efficiency of the OX-phos system is intimately linked to the mitochondrial function. OX-phos stands for oxidative phosphorylation. Primary functions of mitochondria are the synthesis of metabolic water, ATP generation, and heat production. This is an image of a liver cell that reflects a mitochondrial density that is more accurate than most figures that draw the cell's structure. Capturing mitochondria's intricate and dynamic structure poses a challenge as these organelles are very sensitive to light. This is an image of a time-lapse nanoscopic recording called live cell stimulated emission depletion microscopy, also known as STED. They gave the mitochondria a gentle inner membrane stain so you can see the shape and function. Well, not function. On this set of images, on the left side of the slide, you can see more STED images. The top is of mitochondria before and the bottom after deconvolution. This is a process to improve the resolution and reduce the signal-to-noise ratio relative to the acquired image. The middle image, they stained the inner membrane green, and on the right, they stained the outer membrane purple. In mammals, the OX-phos system in mitochondria is composed of 3 types of proton pumping complexes, electron carrier pools, non-proton pumping complex 2, and ATP synthase. The proton pumping complexes 1, 3, and 4 form a supercomplex as imaged here on a cryo-electron microscopy of a porcine heart. They obtained a near atomic resolution structure providing new insights. Complex 1 transfers 2 electrons from NADH to ubiquinone and simultaneously pumps 4 protons from the mitochondrial matrix to the intermembrane space in a single cycle. Complex 3 transfers 2 electrons from ubiquinol to 2 cytochrome C molecules while pumping 2 protons from the matrix to the intermembrane space. Complex 4 delivers 4 electrons from 4 cytochrome C molecules to 1 oxygen molecule, forming 2 water molecules and pumping 4 protons into the intermembrane space at the same time. Note, this is where we synthesize metabolic water. Depending on how these supercomplexes assemble, the structure can increase the electron transfer efficiency and reduce production of reactive oxygen species. Here you can see how the complexes are situated in the inner mitochondrial membrane. This illustration shows the organization of the membrane. The cristae are folded towards the matrix. The Micos complex, which is the mitochondrial contact site and cristae organizing system, sits at the cristae junctions and is composed of several subunits. Cristae were considered to be largely static entities. However, mitochondrial dynamics have demonstrated that these can change shape in a matter of seconds. I want to focus your attention to ATP synthase at the bottom of the loop at the edge of the cristae. ATP synthase is a dimer structure, meaning made up of identical or similar units, and is also known as complex 5. This is another view. ATP synthase is a proton-driven engine, if you will. These subunits rotate. More on that later. This is another depiction of the structure and position of the ATP synthase. These rows of dimers along the cristae are ubiquitous in all mitochondria across species. I briefly want to touch on a few of the theories that are often repeated as the causative factor of mitochondrial dysfunction. These include genetic mutations in nuclear DNA, genetic mutations in mitochondrial DNA, declining NAD levels rather than a lack of low deuterium foods that regenerates NAD+, a lack of hermetic stress weakens the mitochondria, cell danger response signaling shuts the mitochondria down as a protective mechanism. What all of these fine researchers have missed is the kinetic isotope effects of deuterium. Let me explain. What does water have to do with it? In 2021, a team at NASA published a paper called Water and Life, The Medium is the Message. It is a fascinating paper as it details the absolute importance of water in living systems. A few of the quotes that really bring this home include, water defines the chemistry of biology and it is the most frequent and dominant chemical actor in metabolism. Chemical flux shows that a given water molecule frequently and repeatedly serves as a reaction substrate, intermediate, cofactor, and product. Between a third and half of known biochemical reactions involve consumption or production of water. We define water as the chemical cornerstone of biological processes and challenge the notion or even the possibility of an inert biological solvent. Essentially, all biological molecules, large and small, are products of or substrates for biochemical reactions that transform water. This illustration shows the chemical transformations of water by the citric acid cycle. The blues are the protonated water molecules, red, the unprotonated, and green are the water molecules that mechanistically are involved in the reaction. There's water everywhere. Often this is not mentioned in any of the textbooks. So this paper is interesting. As in many papers, they need deuterpretations. And in one of those sections here in the last paragraph of the introduction states that notably our quantitative demonstration of water as the dominant reactive species in biology is distinct from previous defined metabolic water. Mellanby, 1942. That's what they refer to as metabolic water. This is that letter to the editor written by Mellanby, and in it, Mellanby reports that as a result of consuming 100 grams of fat, you produce 110 grams of metabolic water. And as a result of 100 grams of carbohydrates consumption, you produce 55 grams of metabolic water. So if this metabolic water is not produced from low deuterium fat in the mitochondria, you will likely experience health challenges. Health of the mitochondria is ultimately a question of how you interfere with metabolic water production at which step, I should say. So the authors of that paper, they are not aware that all water produced by mitochondria is metabolic water, and that's the water they discuss in all of the reactions that they detail. Hydrogen is the most abundant element in the universe. pH is an arbitrary scale of the power of hydrogen or the potential of hydrogen to react. A hydrogen atom has a proton and an electron. A hydrogen ion is a hydrogen that is stripped of the electron. We call this a proton. The term proton is a Greek word that means first. Ernest Rutherford first used the term in 1920 to describe the nucleus of hydrogen. A proton has charge, size, mass, and spin. And it's a subatomic particle found in the nucleus of every atom. And a proton has a positive charge. Proton tunneling is the energy and information transfer that is essential for life. Nobel laureate Albert St. George stated that life is about recycling protons. The more effectively these move, the more efficient our bodies function. Deuterium affects proton dynamics. Deuterium is the stable isotope of hydrogen. The only difference is a neutron, making it twice as heavy and twice as large. 3 decades of basic research has confirmed the importance of deuterium in living organisms. The ratio of the two stable hydrogen isotopes, hydrogen and deuterium, is a key signal influencing cellular processes at the submolecular level. The transfer of hydrogen and deuterium to different structural and functional molecules is essential for maintaining normal cellular functions, nucleic acid structure, and protein integrity. The term deuteronomics was coined by Dr. Laszlo G. Boros in 2019 and first introduced at the 4th International Congress on Deuterium Depletion in Budapest. Deuteronomics is an interdisciplinary approach to medical biochemistry as it incorporates submolecular isotope effects known about deuterium into clinical reasoning. It is the science of how water moves in our bodies. We all know that water is H2O, 2 hydrogens and 1 oxygen atom. Semi-heavy water is HDO, 1 hydrogen, 1 deuterium, and 1 oxygen. Heavy water is DDO. There's no hydrogen, 2 deuterium, and 1 oxygen. This is very rare. You may have heard of heavy water in the context of it being used in nuclear power plants to slow down the reaction speed. Light water has reduced deuterium content, natural or through column distillation. Metabolic water is deuterium-depleted water our cells produce in mitochondria at complex 4. This is also known as interfacial water or structured water. All water on our planet and outside contains deuterium. The natural abundance of deuterium in oceanic water is 155.76. Most drinking water in the US is between 148 and 152 ppm. Spring waters can be as low as 140 ppm. Wellness water we consider to be 125 ppm. Therapeutic water starts at 105 parts per million. It is believed humans evolved when levels were around 130 parts per million. Maintaining tissue levels of deuterium below 130 ppm will likely prevent mitochondrial dysfunction. Properties of deuterium alter the way a molecule behaves whenever deuterium is part of it. So deuterium makes water heavier. It is twice the weight and size of hydrogen. Deuterium atoms change the temperature that water freezes at, thaws at, and boiling temperatures. Deuterium atoms makes water more viscous. Deuterium is a growth factor for bacteria and yeast, and DNA and RNA viruses depend on it. Deuterium is a growth and transforming factor, acting as an oncoisotope in mammalian cells that have mitochondria. So as I said, once a deuterium atom has replaced a hydrogen, this changes everything of the atom the water molecule reacts with. So for example, bonding affinities and strengths are altered. This is what we call the kinetic isotope effect. Hydrogen bonds split much easier than bonds formed between a deuterium atom and carbon, or nitrogen, or oxygen. This is very unique in the isotopic realm, as there is 100% weight change between hydrogen and deuterium. If we look at carbon, 12C is only 1.04 times faster than the corresponding 13C reaction, even though in both cases the isotope is 1 atomic mass unit heavier. But there's only an 8% weight change between carbon 12 and carbon 13. The carbon-hydrogen bond versus a carbon-deuterium bond is 7 times harder to break. For nitrogen and deuterium, it's 8.5 times harder, and for oxygen and deuterium, it's 10.6 times more difficult to break the bond. So to provide some context for the significance of this ratio, the concentration of deuterium in our body is 2 to 4 times higher than the normal amount of circulating blood glucose. Deuterium is present 5 times higher than calcium and 10 to 18 times that of magnesium. Some more numbers to provide context. We have approximately 35 trillion cells in the human body. On average, 1,000 mitochondria per cell. There are approximately 320,000 of those ATP synthase nanomotors or pumps in one single mitochondrion. And these spin, as I mentioned, they are rotary engines. They spin and release approximately 10 protons per rotation, and they rotate approximately 9,000 times per minute. This is as fast as a Ferrari engine. And it amounts to 13,500 protons released per second that are transferred. So if we think of water that is at 150 parts per million, meaning out of every million hydrogens, there are 150 are deuterium. And if we calculate that, then every 6,600 protons will be 1 deuteron. And at the speed at which these are released, and at that concentration, a deuteron would break a nanomotor every half a second. And this, of course, is why the deuterium-hydrogen ratio is so critical. Deuteronation, may slow down electron transfer in the electron transport chain by interfering with this proton-coupled electron transport reactions, and it increases free radical production, and it breaks down mitochondria. Deuterium changes cholesterol and therefore also vitamin D synthesis. Again, when the deuterium is part of a structure of a molecule, it changes the way it functions. So I remember how, for instance, if we look at cholesterol, it has 27 carbons and 46 hydrogens, and when the sunlight shines on our body and starts resonating the bonds between the hydrogens and the carbons, these start to carry this energy, and there's this movement, and the movement and energy of the photons that are transferred to those bonds actually breaks open the bond between carbons 9 and 10, and that is when you form vitamin D. Now, when there's a deuterium atom attached to that carbon, this structure will not break open, so you will not produce vitamin D from that, and just have circulating cholesterol rather. The same goes for the deuterons that attach themselves to the deoxyribose sugars that form the backbone of DNA. When these deuterium atoms populate this backbone of the DNA helix, it alters the genetic stability and the DNA replication and the repair functions. And this can, of course, give rise to altered gene expression, and there's no genetic variation or susceptibility needed. This can explain the failure of many genomic therapies. Deuterium causes a frame shift mutation and epigenetic changes without any mutations. So let me briefly cover the definition of the TCA cycle, glycolysis, and phenotype. So when I mention these next, it'll be clear what these are. The TCA cycle is the chemical conversion of carbohydrates, fats, and proteins into metabolic water, carbon dioxide, which also generates energy in the form of ATP and heat. And glycolysis is the pathway that we use by all of our tissues for the breakdown of glucose to provide energy and, again, intermediates for other metabolic pathways, including the TCA cycle. Phenotype is the observable characteristics of a trait or an organism, for instance, a healthy cell or a cancerous cell. Phenotype and adaptation depend on mitochondrial metabolism. It is the health of mitochondria that determines the health of the cell, of organs, and of the whole body. The health of mitochondria depends on its ability to produce deuterium-depleted or free metabolic water, and the role of the TCA cycle is to keep deuterium out of the mitochondrial matrix. This primarily happens in glycolysis via isomerization reactions and the TCA cycle through water exchange reactions, providing you have intact mitochondria. So fractionation is the separation of deuterium from hydrogen. This replacing of deuterium with a hydrogen from the cytosolic or matrix water is what we call fractionation. Many biochemical reactions function specifically to protect mitochondria. The reversible isomerase reactions of glycolysis potentially replace all deuterium oncoisotopes that reside on the carbon atoms and in the hydroxyl groups. The structured water pool of the cytoplasm can thus be considered as a deuterium scavenging water tank. The deuterium gets dropped off in there and the hydrogen gets picked up, reattached to the molecule. And that this is how it absorbs deuterium from carbohydrates and amino acids. And the main purpose of the TCA cycle is to transfer hydrogens from carbons to oxygen and back. Mitochondrial dysfunction results when glycolysis can no longer deplete deuterium, and the TCA cycle is unable to maintain these depleting functions. Another essential depleting biochemical pathway is the urea cycle, and peroxisomes are another depleting cellular organelle. So there's 8 enzymatic reactions in the TCA cycle, and 3 of these reactions add deuterium-free water to TCA cycle intermediates from water that is produced in the mitochondrial matrix, also known as metabolic water. Citrate synthase is the first of the three enzymes involved in these water reactions, and it adds one water molecule to form citrate from oxaloacetate and acetyl-CoA. The second enzyme is aconitase hydratase or aconitase. This is a two-step reversible reaction and involves dehydration followed by hydration. This is a water exchange reaction. One water molecule is removed and replaced with a deuterium-depleted water molecule from the matrix. The third enzyme involved in adding water is fumarate hydratase, which catalyzes the reversible hydration of fumarate to malate. If fumarate hydratase is deficient, submolecular dysregulation sets in, causing mitochondrial dysfunction. If the depleting functions of mitochondria are defective or overburdened by any number or combination of factors, submolecular dysregulation and mitochondrial dysfunction are the result. Most mitochondrial dysfunction is due to the lack of oxygen because of damage to or glycosylation of circulating red blood cells, COPD, aging, underlying metabolic disease like diabetes, obesity, and deuterium overloaded mitochondria in all kinds of cells and tissues. Ultimately, natural food plus oxygen plus light equals metabolic depleted water, carbon dioxide, ATP, and heat. Lack of oxygen directly affects the amount of metabolic water produced in mitochondria. Deuterium destroys proton tunneling. Hydrogens stop moving via tunneling and they don't deliver energy through the nanomotor movement. Protons cannot get into the matrix and hydrogens pile up outside the mitochondria, still residing on lactate, leading to extracellular acidification as the water piles up. This is called hydropic degeneration. This is metabolic crowding. This is the Warburg effect. Recent advances in metabolism allowed us to follow the precise metabolic flux or movement of precursors and substrates. Using tracer-based metabolomics, important mechanisms and pathways have been identified. Metabolic profiling involves labeling common precursors, such as glucose, lactate, or acetate, with an isotope so that the pathway this molecule travels can be traced. They use mass spectrometry and stable isotopes such as carbon-13 or deuterium to observe and trace metabolic pathways for gluconeogenesis, glycogen synthesis, lipid synthesis, pentose and TCA cycle metabolism, among others. Restoring submolecular dysregulation. The majority, if not all, of the body's excretory pathways all deplete deuterium for us. Human breath is deuterium depleted in healthy people in the range of 130 to 135 ppm. It depletes oxygen tensions, dilating blood vessels, and as a result, there is more efficient oxygen delivery. Breathing controls metabolic water production through dissolved oxygen in your plasma. Oxygen that is bound to hemoglobin and also by carbon dioxide that is released through biological oxidation. We secrete excess deuterium into saliva, urine, tears, sweat, and stool to release it from the body. A 2019 Nature paper called Meta-omics Analysis of Elite Athletes identifies a performance-enhancing microbe that functions via lactate metabolism. It demonstrated that there is a lactate fermenting atypical bacteria called Veillonella in the gut that produces the shortest fatty acid propionate for de novo fatty acid synthesis from circulating lactic acid in elite athletes. So it functions as a deuterium depleting performance enhancing microbe. That particular species metabolizes lactate into the short chain fatty acid and propionate. One of the main functions of our gut flora is to deplete deuterium for our metabolism. cell proliferation, and pathogenic gut flora propagation or microbial growth requires deuterons, not protons. This flowchart shows some of the key factors that play a role in the accumulation of deuterium levels in our bodies and the effects that they have. Water quality and quantity, fat quality and quantity, and light spectra are the primary inputs that determine deuterium levels. Omnipresent glyphosate, electromagnetic frequencies, and inorganic metals affect submolecular regulation as well. Lack of sulfur and essential trace minerals further impair function and reduce metabolic water synthesis. An imbalance in all of these causes changes in the microbiome, allows for viral and microbial hosting. Remember, deuterium is a growth factor for these organisms. It causes inflammation, it alters vitamin D and A metabolism, causes genome transformation, and metabolic crowding. Submolecular dysregulation due to accumulation of deuterium is at the heart of the cellular transformation, causing mitochondrial dysfunction. The approach to reversing mitochondrial dysfunction is to address all those factors we know contribute to submolecular dysregulation. These include, and by no means are limited to, the standard industrial diet based on corporate agriculture, inferior quality oils, in particular soy, corn, canola, vegetable and seed oils, as well as an imbalance in the ratio of fatty acids, inferior quality of animals and vegetable protein foods and meats that contain synthetic growth hormones, GMOs, antibiotics, dyes, chemicals, you name it. Lack of spring water that is low in deuterium, as well as excessive hydration with fluids that are high in deuterium. Impaired mitochondrial function due to genetic variation in the genes for mitochondrial enzymes. Insulin resistance and metabolic syndrome. Dysregulated iron metabolism, a lack of and dysregulation of important minerals such as calcium, iodine, magnesium, sulfur, copper. Microbiome imbalance presenting as a lack of diversity and an increase in pathogenic microbes. A lack of sunlight and spending time in nature. Night shifts and artificial light at night. Thyroid dysfunction, stress leading to increased cortisol, increasing blood glucose, lack of exercise or movement, increasing age, excessive ionizing radiation and non-ionizing radiation, and this includes ultrasound technology. Electromagnetic radiation, glyphosate replacing glycine, inhibiting many biochemical processes, environmental toxins, plastics, heavy metals, excess alcohol intake, dental procedures such as root canals and amalgams, biologics and mRNA and DNA-based therapeutics, history of oral contraceptives or hormone replacements for menopause, history of steroid use, lack of natural infections of childhood diseases. These are just some of the factors that contribute to the dysfunction. So to reverse this dysfunction, we start with food. Consume a natural, seasonal, and local, low-carbohydrate, well-raised, meat-based, or ketogenic type diet that matches your heritage and preferences. Always choose the best available sources for all the food that you consume as unprocessed as possible. Vegetables should be pesticide and herbicide free and low in oxalates. Foods low in deuterium can be divided as follows. Fats as unprocessed as possible from animals and plants. Animal protein from animals raised in their natural habitat and fed the diet that's appropriate for them. Green and colored vegetables that are low due to the process of photosynthesis, which in itself is a depleting process. Here I have listed some of the foods we measured in the last 2 years as I'm working on a cookbook It's important for us to compose meals that consist of foods that are low and some a little bit higher to kind of balance them all out. And as you can see here, the fats are the lowest that we measured in this set. And this is across the board in all testing that has been done. If we look at proteins, of course, an egg yolk is much richer in fatty acids, and that is lower in deuterium. The egg white is almost 20 ppm higher. And then we have some really great sources of ground beef and pork and lamb and salmon. All of those are on the lower end of the area of proteins. Vegetables, celery is actually quite high at 147. Romaine is 141.8. We measured some fruits here. Those were above 150. And then some other foods that we were interested in: sourdough bread, coffee. We actually also tested an espresso coffee pod, which was 170 ppm. And then cheeses, which are an important part of a Dutch person's life. So those were all around 130. And dark chocolate is 142. The importance of the quality of the food cannot be overstated. Grass-fed meats and dairy are much lower in deuterium content compared to grain-fed animals derived meats and dairy, as shown in this 2022 metabolomics paper. So to facilitate cellular reprogramming and reverse submolecular dysregulation, which will increase metabolic water production and bring the body back in balance, we have created a success list of aspects of daily life to focus on and integrate one by one where needed and possible. So consider your water, the deuterium content, the quality, and the quantity. The foods as discussed. Consider fasting, both intermittent and with clear liquids. Consider the light environment of all the spectra and intensities, including photon biomodulation, sunlight, UV light, infrared light. Try to adhere to circadian rhythms. Include movement, acupuncture, massage. Consider temperature flux, including soaking in enhanced cold water baths and sauna. Electromagnetic frequencies mitigation is very important. Optimize your sleep and your sleep environment. Sleep in darkness. Enhance your oxygen environment. It's very successful to do that with algae and magnets, and try different breathing techniques. Realize that oxygen is one of the two rate limiting factors of metabolic water production besides protons. Add deuterium scavenging botanicals if needed. So these are botanicals that assist the body to deplete deuterium in different ways, in different pathways. Metabolic profiling with 13C glucose tracers experiments have elucidated these details. Botanicals balance the flux. They are adaptogenic. They preferentially bind hydrogens versus deuterium, and the scavenging is based on that increased affinity. And so in a dose-dependent manner, plants can actually decrease the rate of the nucleotide precursors for the ribose synthesis. They can decrease protein synthesis, fatty acid synthesis, and they can decrease the rates of glycogen turnover. The flavoproteins transfer hydrogens. So this is a large class of oxidizing enzymes, and again, they preferentially bind hydrogen over deuterium, and they are essential for maintaining mitochondria by supplying them with deuterium-depleted protons. These are ubiquitous and versatile, and they contain either flavin adenine dinucleotide, FAD, or flavin mononucleotide, FMN. And they restore NAD or NADP plus to NADH or NADPH, and FAD to FADH2. The hydrogen in the NAD is almost never, the hydrogen in an NADPH or an NADH is almost never a deuteron. The reaction will fail. So the proteins will provide deuterium-depleted protons to mitochondria for fatty acid and nucleotide synthesis. And the flavoproteins are derived from riboflavin, a product of the shikimate pathway. Shikimate pathway also produces tryptophan, the precursor to NAD. And glyphosate suppresses this pathway, and thus glyphosate suppresses deuterium depletion and mitochondrial function. Luteolin is one of the flavonoids that has many beneficial properties due to its ability to provide low deuterium hydrogens to regenerate NAD+ and FAD. Quercetin is another, and this is particularly high in onions and cilantro, citrus fruit, apple, pectin, and raspberries. Olive leaf is another. Look at the structure and all the possible binding sites for hydrogens. The embryonic plant extract of olive leaf contains luteolin, quercetin, and many others, and has many beneficial properties. This is a nutritional label of a popular NAD supplement. Note an NAD plus supplement. Notice that this is a deuterium depleting formula. It has the nicotinamide mononucleotide and all of the botanicals I just discussed. Foods high in niacin, the precursor of nicotinamide, are meats, fish, poultry, as well as avocado, mushrooms, and green peas. Deuterium depleted water. There is no question that deuterium depleted water can reverse submolecular dysregulation very rapidly and efficiently. It can be used as wellness water or as a therapy, meaning the ppm you consume is based on optimal physiological levels and determined based on weight, diagnosis, and consumption level. For those who have been diagnosed with a cancerous process, adding deuterium depleted water to other therapies has shown to increase overall survival time 3 to 5-fold. 11.1 years, or 4 to 5-fold increase. That's the data that was reported by Gabor Shumiaye. This is based on 204 tumor-free cancer patients. 159 repeated deuterium-depleted water treatments. They had no recurrence at all. There were 2 deaths in that group, but neither was cancer-related. 22 patients did not repeat the treatment, they just did it once, and there were 8 deaths that were cancer-related in that group. The cycling in and out of periods of consumption of the DDW, the deuterium-depleted water, of varying parts per million is beneficial. Integration of deuterium-depleted water into the conventional therapies can drastically reduce the number of cancer-related deaths. Start DDW as close as possible to the diagnosis and consume the proper dose. This may prevent recurrence or relapse. This is all the data that was reported from this particular retrospectively a study that was published based on Hungarian population data. So the mechanisms of deuterium depleted water are many, and they have been published in the research. And here's a list of these. So it inhibits cell proliferation. It prevents the ratio from increasing to a threshold that is required for cell division. It induces cell cycle arrest and apoptosis and increases autophagy. It can selectively stress tumor cells, but not healthy tissue, and it decreases the tumor growth and metastasis rate. It restores the activity of SOD, and it restores mitochondrial fumarate hydratase activity. Further papers include that it prevents the expression of cancer-related genes, and it reduces the number of single-stranded DNA breaks, decreases expression of specific genes that are important for metastasis. Depletion protects cells by maintaining strong hydrogen bond networks. It's radioprotective and it improves thermoregulation. It's anti-inflammatory. So where do we go next with all of this incredible information? The future directions include standardizing the MRI imaging. So, as I said, a proton has a spin, and that's the spin what we can actually view on magnetic resonance imaging. A deuteron changes the spin of the hydrogens around it or the protons around it, and so coming up with ways to standardize this is and hopefully in the future that we can very specifically show you where deuterium is in tissue. We would like to increase the testing availability both in the US and in Europe to test the patients and realize so that we can check therapies, know where we are, and maintain optimal conditions. Deuterium of existing research and data is a really important aspect because much important research has been done, but it just has not been correctly interpreted. And so this is something to work towards. Another very important aspect is identifying the precise mechanism by which submolecular regulation is governed. And I will talk about that briefly in this new paper that was published last year in relation to that. There are even more fascinating areas to cover in deuteronomics, and that has to do with the scriptures, interpretation, and ancient history. So as I said, this fascinating original paper came out in early 2022 by Roman Suborov from the Karolinska Institute, and it's called Abnormal Hydroxyproline Deuterium Content Redefines Hydrogen Chemical Mass. So there's 21 amino acids that make up proteins, and of those there are a few with very unique property that gives them the ability to bind deuterium. If you look at collagen, there is glycine-proline pair of residues. Glycine is the most simple, and proline has a 4-carbon ring structure to which either hydrogen or deuterium is bound. When there's deuterium, that increases the bonding strength. Hydroxyproline can also proline can also carry deuterium and further strengthens collagen. Just to reiterate how the strength of the bond impacts these tissues, when a deuterium ion is bound to nitrogen, the bond is 15 times harder to break. Deuterium bound to oxygen is 10 times harder to break. When deuterium is bound to carbon, it is 7.5 times harder to break. We know that 25 to 35% of the amino acids in collagen are proline and hydroxyproline, and thus form the majority of amino acids in collagen. In biology, the structural proteins are strengthened by deuterium, while the moving proteins are destroyed by deuterium. There has to be a biochemical pathway that sorts these two ions out, hydrogen and deuterium, for various biological purposes. That's why those involved in deuteronomics research, are trying to solve for now. What is this pathway of elevating deuterium above its natural enrichment? And this is an important piece. So this paper took a look at the bone of different animals. They looked at the Curver's beaked whale, they looked at an elephant seal, a peregrine falcon, the gray seal, a harbor seal, a polar bear, a sperm whale, and a swan. And what they found is that the highest level of deuterium, which was much above natural abundance in nature, was over 300 ppm. This is extremely unusual because there's nothing the animal can eat of that strength. So somehow there is a mechanism by which the gray seal places the excess deuterium in the bone to provide strength. It dives really deep, so it has to be able to withstand the pressure of the water. So it's fascinating. They looked at the other animals, and they were not nearly as close as the gray seal. So long story short, mitochondrial health is essential for bone strength, and of course we want to understand how this works in light of prevention of fractures and osteoporosis. Um, it's for many reasons a very important aspect of trying to figure out what regulates deuterium into what tissue and how can we lead that into the right places. Because, of course, some molecular regulation of deuterium is the key here. So this is the whole other area that's utterly fascinating, and a lot of work is being done in the backgrounds of trying to interpret with religious researchers and specialists to understand how we can interpret the scriptures. There's ongoing discussions, and hopefully these at some point will provide new insights into deuteronomics. In 1961, there was a set of discs discovered by archaeologists in Romania, and these have been interpreted by Dr. Laszlo Boros, and he has described the images on there as relating to deuterium, and it's another absolutely fascinating exploration that at this point has mostly been done in Hungarian, and hopefully in the future we will be able to translate all of this as well. But these discoveries beg the question whether ancient cultures were way ahead of us and knew about deuteronomics. And some scientists propose that these tablets date back to around 5300 BC. So that predates a Mesopotamian pictographic proto-writing. So again, Dr. Boros has presented on the interpretation of these discs in Hungarian, and together with historians, they continue to interpret the symbols. So all human qualities, abilities, and prophecies are dependent on depleted ketogenic metabolic states of sacrifice animals and humans who sacrifice them with a properly functioning microbiome that separates the high deuterium sugary environment from cells, mitochondria, and metabolic water production. Laszlo Boros. In summary, mitochondrial dysfunction is caused by excess deuterium that disrupts proton tunneling, slows down the electron transport chain, and destroys the nanomotors of ATP synthase. If the depleting functions of mitochondria are defective or overburdened by any number or combination of factors, submolecular dysregulation and mitochondrial dysfunction are the result. Thank you.